Hello everybody, Sipati here and welcome to the Gemophiles Microbiology in a Flash. From previous videos, we now know what prokaryotic and eukaryotic cells look like, and we know what the different components within these cells do. How then do these cells ensure their survival and longevity? Well, one of the most important ways of doing this is by multiplying. We limit the likelihood of our extinction by reproducing and growing our families. So, microbial cells can also multiply to increase their populations and ensure their survival. In today's video, we will discuss how microbial cells divide. Microbial cells divide by binary fission, which is the only method to reproduce new individuals in unicellular microbial cells. Binary fission occurs by the division of the mother cell into two identical daughter cells. It is facilitated by the FTS proteins, which form part of the prokaryotic cytoskeleton. These proteins will be discussed in a future video. Binary fission starts with the replication of the genetic material carrying hereditary information, for example, the circular chromosome in prokaryotic cells. The chromosome is replicated first at the origin of replication. This is the point where the DNA double helix opens up in preparation for replication enzymes to attach and begin the replication process. The two copies are then pulled to opposite ends of the cell. The cell then extends its length. DNA replication continues until the entire chromosome is copied and the copies are at opposite ends of the elongated cell. The duplication of the genetic material is known as karyokinesis. The next step is for the cytoplasm to divide in a process known as cytokinesis. This starts with the ingrowth of the cell membrane, and in cases where cells have cell walls, the ingrowth of the cell wall in the middle of the cell, which forms a wall that separates the two new chromosome copies. This wall is known as a septum. Once the septum is fully developed, it then separates, and what was once a single cell now becomes two cells. And that is how microbial cells divide. That's it for today. See you guys in the comment section or in our email inbox. Thanks for watching and see you next time on the Gemophile.